you, Andreas. First of all, I'd like to thank the FAME Lab for the invitation. Our topic is going to be how we can use the oxidative stress markers to monitor sports-related maladaptations, but I'm going to use the overtraining model, which is the most famous one among all these sports-related maladaptations. In the first part of my speech, I'm going to introduce you to the overtraining concept. Then I will try to convince you that there is a link between oxidative stress and overtraining. And at the end, we are going to talk about about the criteria, if oxidative stress markers actually fulfill the criteria as diagnostic markers for overtraining. Okay, you're all familiar with the training model. We have a training session, and then we have acute fatigue, okay? It's physiological, it's normal for an athlete to get tired due to the training stimulus. However, we're not going to talk about one training session. So we're going to talk about multiple training sessions. And as you can see here, between training sessions, we have a period which is called recovery, during which athletes are trying to recover metabolically, neuromuscularly, and in general to be able to perform in the next training session. And this goes on and on. Our goal is to increase performance at the end of the training period. In this model, training and recovery are well balanced, okay? And as you can see, we have no imbalances. And you are all familiar with the periodization model, which basically tries to adjust the training overload in order to have peak performance and at the same time avoid injuries and maladaptations. But this is not always the case, okay? Periodization sometimes works, sometimes doesn't. In this model, we have, again, multiple training sessions, but as you can see, the training overload is much heavier than the recovery. So we have an imbalance. How big this imbalance is, we'll see. Nevertheless, performance is going to decline because the training stimulus is quite heavy and the recovery is not what's supposed to be. Therefore, we have physiological side effects, and at the same time, we have psychological side effects that accompany the fatigue athletes usually feel. However, in this model, although we have an imbalance, okay, we don't have the side effects, which means it's not that bad, okay? At the end, we're going to have an increase of performance. We may have a decline of performance after each training session, but at the end, performance will increase. This is called functional overreaching, okay? And most coaches and most trainers aim to induce this kind of overreaching. This is a good example. If you see this figure here on the left-hand side, you can see that we have athletes that followed this kind of periodization model. We have a 3 microcycle scheme where the overload increases progressively, and then we have a taper of 2 weeks. Okay? When they did this study, they tried to see if the increase in overload will cause overtraining, will cause some kind of negative side effects. However, when they used this model and the athletes participated in the World Championship, as you can see, it worked perfectly because we had this group had 4 gold medals and 1 silver. Okay, this is a 3 microcycle up, and then we have a taper of 2 weeks. Therefore, it was a functional overreaching. okay? What every coach wants. However, we have another case where the imbalance between training and recovery is quite heavy, is quite big, large. And then we have physiological side effects and psychological side effects. And recovery needs to be a little bit longer. And this is non-functional overreaching. And here we have a problem due to these side effects. So what about performance? Is it going to recover? Yes, it's going to recover, but it's going to take longer, okay? In this case, however, things are a little bit worse, okay? Because we have the training stimulus here. We do have recovery, but recovery is not what's supposed to be. And at the same time, we have an intense competitive schedule. For example, too many matches during the season. We do have malnutrition, not good nutrition habits, okay? We do have psychological pressure by the coach or by the teammates, by the fans. We do have biomechanical problems, poor technique. We may have also illnesses, okay, which is a side effect of overtraining. We do have other environmental factors affecting the whole thing. We do have financial problems, as we may have this kind of stressor, which is not that strange in our country nowadays. And the athletes may not sleep that well, and also they also lost the motivation due to all these things that happen. And of course, social problems could be present. All this, and of course bad psychology, and all this will induce what we call overtraining, which means that it's not only the training stimulus that affects things, but it's also a number of stressors that come into play. Physiologically, we need to follow this paradigm here, which means that we give a stimulus, an intense stimulus, the performance declines only temporarily and then goes up, and then we give another stimulus, another training stimulus, it goes down just for a short period of time, and then goes up and then goes down. At the end, it's going up, okay? But in this model, we give the training stimuli, and at the end, it goes down. This is what we don't want to happen, okay? And overtraining is about more training stimulus and less recovery. And in 2012, 2013, the most recent report on overtraining was published by a joint paper, in a joint paper written by both American and European sports scientists. And they ended up defining overtraining like this, okay? And they are talking about training and non-training stress that combine to create all these side effects that ultimately lead to a decline in performance. However, overtraining is a clinical situation. It's a clinical situation because it has side effects. It has a pathophysiology, which means that the immune system is affected, the cardiovascular system is affected, the musculoskeletal system is affected, and this is not what we want from our training. And as you can see, functional overreaching is okay. However, when we proceed to non-functional overreaching and overtraining, things are a little bit puzzling. And sometimes coaches and physicians are not able to distinguish between these three stages, okay? And why they are not able to distinguish between these three stages is because we don't have very sensitive biomarkers, very sensitive diagnostic tools in order to monitor this process. And as you can see in this slide, the epidemiological data, the epidemiological evidence is quite strong. And they say that most of the athletes, a large percentage of athletes, experience overtraining at least once during their career. So It's not something that we can take lightly. So we have to answer a few questions. Can we monitor overtraining? Do we have markers to actually say we do have an overtrained athlete, or he has some kind of risk factors that predispose him to overtraining? That's what we are going to talk about. The reason I came up with this presentation is why during the last paper published, position statement that was published on overtraining syndrome, these guys here, which are very well-known, distinguished sports scientists, they talked about a large number of overtraining markers. At the end of their position statement, they state that at the moment, we do not have a good overtraining marker. Sensitive, valid, and reliable, except the unexplained underperformance, declining performance that we cannot explain, which persists for a long time. Therefore, we don't have biological markers to monitor this phenomenon. Here are some of the markers that have been used in the past in the literature. And you can see the most valid one is performance testing. Okay? And this is the actual criterion. If we reduce the overload, the training overload, and the decline performance persists, then we do have overtraining. And if it persists for a long time, things are even worse. However, most of the biochemical markers, either metabolic, hormonal, immune markers that have been used, were not good enough to systematically monitor the overtraining. Okay? And these are all markers that have been used. This one is a questionnaire, a questionnaire on psychological side effects. Okay, it's called POMS and has been used consistently, and it has given some valid results, because overtrained athletes usually experience either aggressiveness, aggressiveness, fatigue, hypersensitivity, or complete apathy. It has been shown that this kind of questionnaires may be able to pick up this kind of side effects. Okay, and it has a good, from all the markers already used, it has the highest validity and reliability. Physiological markers, immunological markers, and so on. I have to say here that Dr. Meusen, from Belgium, has developed a test with 2 consecutive exercise tests on the bike or on the treadmill, separated by 4 hours. And he measures hormonal responses, pituitary and hypothalamus hormones, before and after these 2 tests. He has published some results that are a little bit promising, but not conclusive yet. Okay, so what's the connection between oxidative stress and overtraining? If we want to establish a hypothesis that oxidative stress biomarkers are helpful in monitoring and diagnosing overtraining, we have first to prove that there's a connection. Professor Djamourdas just a few minutes ago explained all the biology of free radicals. And by now, you are very familiar with them. Okay. First of all, we have to see what's going on. After each intense training session, especially if this training session includes a large number of eccentric contractions, we do have muscle damage. The higher the stimulus, the higher the damage. And due to the damage, we have an inflammatory response. An inflammatory response means that we do have a rise in immune cell recruitment. And mobilization, which means neutrophils and macrophages. These are the two types of immune cells that are recruited during the first hours or days of muscle damage. What happens next? Dr. Jamurdas already explained that we have this respiratory burst, which means that we have a large increase, a large generation of free radicals. Why? Because they need to clear up the debris and all these fragments that are piled up in the muscle due to the damage. And if we want to regenerate the muscle, to heal the muscle, we have to get rid of this debris. That's why we do have the reactive oxygen species, and that's why at the same time we do have a rise in all antioxidants. It depends on the stimulus, okay? The inflammatory response after, for example, football is very, very different compared to the one induced by basketball or handball or a marathon run or a triathlon, okay? It's very different. So what I'm going to try to convey is that we do have a connection between overtraining and free radicals. We do have proteolysis. Why do we have proteolysis? Because a lot of proteolytic enzymes are activated due to the increase in intracellular calcium. At the same time, we do have the free radicals, which are trying to clean up the injured area. And we have seen a large increase of free radicals, of oxidative stress biomarkers after a large number of exercise activities. For example, this one is after Ecentric, intense, very, very intense Ecentric exercise protocol. And as you can see here, we do have a very, very, very increased accumulation of macrophages inside the muscle. Okay, therefore we have seen that. Plyometrics. Plyometrics is, for those who don't know, is just jumping exercises, and jumping exercises has a very, very strong eccentric component. The same thing. We do have muscle damage, and we do have a rise in inflammatory markers. For example, CRP, C-reactive protein, leukocytes, uric acid. And of course, running, just simple running. Intense running can cause inflammation and a rise in oxidative stress biomarkers. For example, protein carbonyls, T-bars, a number of lipid peroxidation and protein carbonylation markers. Here you can see a number of studies showing that after a football match, just a single football match, we do have a very significant rise in all these markers. Basketball, handball, and so on. So after a single event, we do have a rise in free radicals, we do have a rise in inflammatory markers. So we have seen the same things after training sessions, a single training session, and multiple training sessions in our latest work. But what about overtraining? Because overtraining is not about only one match, it's not about only one training session, it's about accumulative training stimulus. Okay, Pedersen from Denmark and Cannon in the 90s have developed a theory which says that when the muscle is damaged down here, it produces signals. And these signals are in the form of molecules called myokines. It's like cytokines, but only produced by the muscle. When these signals are produced, they go to a number of other tissues, such as the central nervous system, the brain, the liver, probably the fat tissue, and they inform these tissues about the damage. So these tissues have to commit to a number of actions that aim to the recruitment of immune cells. And immune cells are recruited and they infiltrate the muscle, they infiltrate the damaged area in order to clean up the damage. So do cytokines increase after an intense exercise? Yes. You can see here data from a marathon race, after a marathon race. They do increase. What does this mean? Cytokines activate immune cells, and immune cells go to the muscle, and what happens there? But Professor Jamoussas just told us that the NADPH activity of these immune cells is an enzyme located at on the membrane, in the membrane of these cells, produce free radicals. Okay, and this has been shown by very, very sensitive and delicate immunohistochemical techniques. And of course, in this type of studies, proteomic studies, Gandra and colleagues showed that a number of pathways are upregulated during the first 2 or 3 days after muscle damage occurs. Okay, and you know, I have to say here that overtraining studies are extremely difficult. Why? Because you can never obtain an approval to perform a study where you will say to the subjects that I'm going to overtrain you, I'm going to injure your muscles, and I'm going to induce some bad things to you in order to study a few things. You can do that. You can't do that. So it's difficult to do an overtraining study. And most of the studies, most of the data come from animal studies. Okay? So if you see here, you can say that we have an upregulation in protein pathways, not only metabolic pathways, but also pathways that are involved with the proteins that actually get damaged, such as the sarcomeric proteins. And as you can see, in animal overreaching, not overtraining, overreaching, you can see that we do have an increase in antioxidant activity and, of course, oxidative stress biomarkers. They train the animals to get a stage of functional overreaching. Okay? Hall and Tal in 2009 published a report. They tried to develop a protocol in order to induce overtraining in rats. Okay, and this is a little bit difficult. We have to confirm that overtraining indeed occur. Okay, and this is not difficult. It's not easy. And as you can see here, this protocol induced a performance decline. But is this overtraining? I'm not sure. Positive adaptations that were induced with previous training were lost. However, this group did not perform any oxidative stress measurements. So we do have a valid protocol for animal overtraining, but we didn't have any oxidative stress measurements. However, research proceeds and advances, and a lot of other groups try to develop a protocol, an overtraining protocol for animals. And as you can see here, this is an interesting study where we do have this protocol that induced overreaching for sure, a significant performance decline after a while. And as you can see here, certain mitochondrial proteins were compromised. For example, you can see here the control stage, the functional overreaching, and the non-functional overreaching stage. You can see here that we have a serious compromise in this kind of proteins. So mitochondrial function declined. And when mitochondrial function declines, we do have free radical generation due to the uncoupling. They did induce overtraining in these rats, in that study, and they did measure oxidative stress markers, and they went all up. Okay, and this is the first study that convinced me that something goes on with the oxidative stress markers. A friend of ours, Professor Radang from Hungary, actually showed something else, that not only overtraining increases oxidative stress biomarkers, but at the same time changes the brain activity for good or for bad? Probably for bad, but the animals were trained and they demonstrated a deviation. And we know that overtrained athletes exhibit either hypersensitivity, aggressiveness, or complete apathy at certain stage. And of course, some other studies that also showed they linked muscle damage and inflammation to oxidative burst activity. But what about humans? Well, we didn't have a lot of human studies. Why? Because of the reason I mentioned before. However, this is a study that Voller did and his colleagues did and actually didn't achieve overtraining. This is overreaching, okay, not overtraining. And then our group comes in, okay, and we did a study with resistance exercise training. And I can explain you, we took the guys here at the baseline, we had them trained for 3 weeks, very low training load. The exercises were multi-joint exercises, not single-joint exercises. Exercise like squats and deadlifts and hang cleans and all these kind of nice things, okay? But the training load was pretty low. The frequency was only 2 days per week. And then we increased 8-fold the overload. And for 3 weeks, we stopped for 5 days. We did some measurements here and here. Then we went to the stage number 3. And here we increased the training load 16 times compared to the first phase. And then we reduced it. It was in this 4th stage, it was equal to the first one. And then we just completely abolished training, stopped training, okay? What we found? We found that performance declined. The performance declined here, and performance declined here. Do we have overtraining? We definitely have non-functional overreaching. Do we have overtraining? I'm not that sure. But we do have functional and non-functional overreaching because we reduced the training load, but the performance did not recover, stayed down, okay? And we did measure a lot of biomarkers. What we found? We found that all biomarkers, all oxidative stress biomarkers went up. This is the shaded area is the phase number 3, okay, with the highest overload. And as you can see here, all oxidative stress biomarkers changed completely. Okay, but the most fascinating finding was this one. Okay, oxidative stress biomarkers went up, but look at the total antioxidant activity in the blood. Went down. In the first 2 phases, went up compared to baseline, but at the peak, it went down. Okay? And actually, I lost my sleep over this because I wasn't sure if results were correct. What if we did the measurements wrong? Okay? So I wasn't certain about this finding because I haven't found, until that time, I hadn't found a similar finding with training. So I was a little bit confused there. However, a group from Finland, Taskanen and Tal, a friend of ours, Professor Atalai's group, did 2 studies about 2 years after ours. Okay, what they found was in this first one, they used soldiers. Okay, in the States they have used soldiers for a lot of this kind of studies, and soldiers are involved in very, very heavy training. So during a very, very heavy training, for 1 week, what they found that all the oxidative stress markers went up, but as you can see here. Something went down. And this something that went down was the antioxidant activity. So that was good for me, okay? My students measured actually the oxidative stress biomarkers correctly in the lab. But the most important study, according to me, was the next one. Because these guys in Finland, they have an overtraining clinic. They have an outpatient overtraining clinic where athletes that believe that they have an overtraining problem, they go there, they go to the hospital, and they say to the doctors, just check me out, okay? So they get to see a lot of overtrained athletes in that clinic. So they took a group of this kind of athletes, okay? And what they saw was that all these athletes who were really overtrained, because they were diagnosed with overtraining, they had increased oxidative stress biomarkers, protein carbonyls, and so on. But the most important thing is that they also showed increased cytokines, pro-inflammatory cytokines, okay? And, sorry, they did see a downslope of antioxidant activity. And basically, it was the second study, the third study, including ours, that they showed that when we reach a non-functional overreaching or overtraining stage, antioxidant activity goes down. Why? I'm not really sure, but ours and their speculation is that antioxidant activity is compromised during this kind of maladaptation. Okay, we do have an excessive generation of free radicals, and antioxidant activity is not able to offset the whole thing. And then we do have some other studies that actually, they showed that mitochondria also contribute to Roche generation during overtraining. These studies. Okay, so it's not only the immune cells that contribute to Roche generation, free radical generation, but it's also muscle mitochondria. Why? Because they get damaged. And one study before showed that. So, are they useful? We definitely have evidence that free radicals are there during overtraining. But can we use them? Let's see. I will pass all these slides because they say more or less the same thing. And I will go straight to the last part where I think it's the most important. What kind of studies do we need? We need studies that they have a sufficient number of well-trained athletes, not untrained individuals, trained athletes who are exposed to accumulative, intensive training stimulus. We need to confirm that these athletes reach functional, non-functional overreaching or overtraining. However, overtraining is a little bit difficult to observe because it's a very long-term process. We need to demonstrate that we have a drop in sport performance. How do we measure sport performance? In a number of ways, depends on the athletes. However, we have to demonstrate that performance goes down despite prolonged recovery. What else? We have to exclude other reasons that could lead to impaired performance. What else? We have to include markers measured both at baseline and after submaximal exercise. In our study, if you noticed, we took blood 5 days after the last training session of each stage. So you need to leave some room and then take blood. And of course, we have to do a follow-up. And if free radicals, if oxidative stress biomarkers are good as diagnostic markers, we need to show that we have a change in these markers towards a certain direction, either up or down, consistently. And in order to do that, we have to show this. And I think this is the most important slide in all this, in this presentation. We provide a training stimulus, a training load. So we do have to monitor performance and the marker in question. Okay? Now, if the training load goes up, we expect performance to improve. Okay? And of course, we expect the marker, our marker, to increase. Okay? It's normal for performance to improve. Okay? However, if we provide even a larger stimulus, what is going to happen? Performance will decline. And what else is going to happen? Our marker is going up, continues to rise. Now, if we cut down the training stimulus, what should we expect? We should expect that performance will remain down. And this is a sign of non-functional overreaching or overtraining. What about the marker? Performance is still down. The marker should be still up. If the marker goes down, it means what? It means that it cannot be used. It normalizes. We need to have an alter response at a stage where the performance is still down. Do we have this kind of response with oxidative stress biomarkers? We do have this kind of response with antioxidant activity. I'm not sure if we do have this response with oxidative stress biomarkers, the rest of them. But total antioxidant activity is compromised. Okay? And these studies actually show that. So do they need a certain attention in the literature? Do oxidative stress biomarkers deserve some kind of attention in the literature? I believe so. Are they easy to measure? Yes, they are easy to measure. Is it cheap to measure? Is it cost effective? Is it a cost effective measurement? Yes, I believe so. I think there are a number of companies right now. For example, I know one in Italy that develop equipment, photometers, that can actually measure antioxidant capacity very easily, very quickly, and without spending a lot of money. Okay? And basically, that's what I explained in a number of criteria. The change should reflect a chronic response, which means that if we stop the training, if you stop athletes training, then the marker should still remain up or down, altered. Okay? And this happens with the oxidative stress biomarkers, as I explained. Look at this. We do have training sessions, and then we have X training sessions. And you can see here, as in the Margonis et al. Study, and they remained altered despite recovery. Do they coincide with changes in other symptoms? So, for example, if we have a change in our biomarker, does this change coincide with other changes, for example, psychological changes, immune changes, cardiovascular changes, musculoskeletal changes? It has to be seen. We don't know. And these are some other criteria, mostly of biochemical nature, but I think from the evidence and data we have gathered so far, that oxidative stress biomarkers may be able to fulfill most of these criteria. So I think in the future we need to bring our attention mostly to try to focus on what happens with the antioxidant status, because it seems to be compromised at the overtraining stage. And I'm not talking about the total antioxidant activity, I'm also talking about the glutathione status. One of the studies showed that when we reach an overtraining stage, glutathione goes down, reduced glutathione goes down. So it means that something happens in the muscle, and the antioxidant system cannot keep up with free radical generation. So it's something worthwhile to explore. However, overtraining is not about biochemistry. Overtraining is about training. And the best way to monitor right now overtraining is by monitoring the training load. If the training load goes up, performance goes down. We cut down the training load, performance still is down, then we have a problem. We have to rest our athletes. If we rest our athletes, and they are still tired, the performance is still down after 2, 3, 4 weeks, we may have a more serious problem. It would be nice to have a single marker that by measuring it, we'll be able to say, yes, we do have overtraining. We do not have this kind of marker right now, but hopefully in the future, I know that a number of labs work on it, and we may do have this kind of marker, magic marker. Open to questions. Of course, I would like to thank these guys here who actually helped a lot. It's our group. It's the group in Komotini, Department of Exercise Science, where I used to work until October of 2015. Of course, Thanasis Tsamourdas and Yannis Koutendakis, who helped a lot with all the studies we did. Some other guys from other institutions, like, for example, Dimitris Kouredas, Michalis Nikolaidis, Mike Strazis, some guys from the medical school in Athens, and of course some guys from abroad who actually helped a lot in a number of aspects of these studies. Thank you very much.